therapeutic approach to blood disorders through CRISPR gene editing.
Before we discuss the critical details of our study, let's go over some background information. With the help of CRISPR gene editor, scientists are working on a way to be able to use this gene editing to alleviate the symptoms of blood disorders. CRISPR is a rising gene editing technology that permits scientists to be able to edit parts of the genome by removing, adding, and altering parts of the DNA sequence as seen on here. CRISPR uses a guide RNA and Cas9 protein in order to remove the target sequence and reattach the new end successfully. This study focuses on people who have sickle cell disease or other types of diseases resulting in lower oxygen levels in the bloodstream. Individuals with the disease have it as a result of the gene that encodes for a mutation of the hemoglobin subunit called beta. Without this subunit being produced properly, oxygen isn't effectively delivered to tissues in the body. However, fetal hemoglobin, which has a much higher affinity for oxygen, depends on gamma subunits being produced. Since sickle cell only affects beta subunits, the production of gamma subunits in adults would help a great deal with the effects of sickle cell, and oxygen can be transported effectively in the body. The gene editing causes a deletion in individuals, allowing them to maintain their fetal hemoglobin levels even through adulthood and therefore not switch to primarily beta subunits. This results in much more proper red blood cell production and no exhibition of the disease's traits.
Prior to experimentation, it was discovered that individuals heterozygous for the wild type and the 13 nucleotide deletion have 30% fetal hemoglobin, which is potentially therapeutic for sickle cell disease, compared to individuals that completely lack the 13 nucleotide deletion or HPFH mutation who produce less than 1% fetal hemoglobin. So to test the effects of hereditary persistence of fetal hemoglobin or HPFH on human blood cells infected with sickle cell diseases, Professor Michael Weiss and his team at St. Jude's focused on a 13 nucleotide sequence as seen on this diagram. The specific 13 nucleotide sequence located on the HBG1 gene is believed to repress further production of fetal hemoglobin on blood cells, thus creating the gamma to beta or fetal to adult shift in hemoglobin populations. So the team reasoned that CRISPR-Cas9 editing followed by error-prone non-homologous end joining or NHEJ to recreate the effects of the 13 nucleotide deleted or HPFH associated HBG1 mutation. So how does CRISPR-Cas9 gene editing work? CRISPR consists of two components, a guide RNA and an enzyme known as Cas9. The guide RNA is a short synthetic RNA composed of a targeting sequence which defines the genomic target to be modified. The Cas9 guide RNA complex binds to the target DNA and eventually creates a double-strand break. The resulting break is then repaired with non-homologous end joining as mentioned before.
The team edited stem cells from 3 patients with sickle cell disease and examined the red blood cell progeny. Then they cultured SCD red blood cells. The control cultures contained relatively high percentages of fetal cells, 65%, although 25% of the cells within the entire population showed sickle cell-like morphology. In contrast, the Cas9 plus gRNA1 expression increased the proportion of F cells to 90% and reduced sickle-like morphology to 4%. Thus, targeting the 13 nucleotide associated region from patients with sickle cell disease inhibited the expression of the phenotype in the culture. One main concern of the study is the common ability of Cas9 to cause a mutation in off-target sequences. Out of 344 colonies tested, 30 had on-target mutations of the specific 13 nucleotide strand, therefore meaning that roughly 8% deleted the exact strand and successfully produced enough fetal hemoglobin.
Here are some applications of this study to our course. The usefulness of producing fetal hemoglobin to alleviate the symptoms of sickle cell anemia has been prevalent in the scientific community at least since the 1980s. CRISPR gene editing technology has evolved from hydroxyurea, which was an oral drug shown to enhance fetal hemoglobin production. The study of genome editing is related to our course content in many ways. First, the Cas9 enzyme associated with the CRISPR gene editing tool is similar to other enzymes involved in DNA replication we have discussed. Cas9 has a specific role of removing a target sequence in which the editing cannot work successfully without this enzyme, just as DNA replication cannot function without its specific enzymes. The study talks about indels, insertion or deletion of a segment of DNA causing a mutation, which is exactly what CRISPR does. We have discussed the deletion or addition of a single nucleotide and the drastic mutations that result from this, which supports the study and that the deletion of 13 nucleotides results in a drastic phenotypic change. Another application of this study is gene expression. Proteins are no longer produced to stop fetal hemoglobin production and switch to adult hemoglobin. Therefore, the blood disorder associated with adult hemoglobin is not expressed.
CRISPR does raise some bioethical concerns since it opens up the possibility of editing parts of the human genome and changing what traits people express. This is much like in the case of Molly Nash, which also created ethical concerns about editing and subsequently changing the expression of diseases and outcomes of certain traits. This study has not been used in clinical trials on human patients, as scientists seek to minimize potentially harmful off-target mutations and compare different approaches to determine which one is safest and most effective.